Hi everybody, I hope you're having a good evening. This is one of my first videos, so please bear with me. Biology 1 Scientific Method. So we're going to hopefully go through this pretty quickly, and you should have enough experience with the scientific method in your past science classes. So I'm assuming you've had a good exposure to this. Scientific method is you're stating a problem. A statement or question that clearly defines what it is you're trying to solve. Make sure that it's testable. There are things that we can't test. I can't test a dog's happiness based on the number of times it wiggles its tail. I can't test to see if a fish is happy. We don't test emotion. We test things that we can more like count, that are not emotional, that are how tall a tree grows based on how much water I feed it. Those are things that in science we can test. A hypothesis should be in the if-then format. You have, if crickets are placed in a cooler temperature, then they will decrease movement. If crickets are, I think crickets will decrease movement in a cooler temperature. Which one of these two is the if-then? Do you see how easy it is? Really, it's the if-then statement is what we're looking for. Okay? And it's an educated guess that you think will happen. Because a hypothesis is something that you think will happen, is there a probability that you're going to be wrong? Yeah. And it's okay. That's what science is all about. So you're wrong. You learn something from being wrong. So in my class, I want you to realize that experiments are going to go wrong, and there's nothing wrong with that. And you're going to learn, hey, well, that didn't work, right? You learned something. So don't freak out on me when things just don't work. Writing a hypothesis. Eels are placed in dark cages and light cages. They are found to have a slower swim speed in dark cages. So what would a hypothesis look like? Has to be an if-then, right? So if eels are placed in dark cages, they are found to have slower swim speeds in dark cages than in light cages. If-then is what you want to write it. Procedures. Written so any person could do it identical, step by step. If I give you instructions on how to do peanut butter and jelly sandwich, I have to get to the point of telling you, go get the bag of bread. Untwist by turning to the left. Stick your right hand in there and pull the bread out. You see how I'm taking you step by step, just like a recipe. You're doing the same thing. If I send this to a kid in Japan, they should be able to do your experiment based on your steps. A lot of students get lazy with that and they decide, I just don't want to write it. Sorry, not science. Got to take it step by step. Got to take that time. The data that we write is it needs to be organized. It's showing you what your measurements are. It's your proof. So yes, there's a lot of math. And of course, we have conclusion, a statement that explains your hypothesis was correct or incorrect. Your actual data from the experiment should be in your conclusion. That's what's backing it up. Now, in making a graph, you have the independent variable and the dependent variable. Your independent, it's what's going to go on the x-axis. Okay? It's the variable you are testing. It is the cause. Type of fertilizer. The fertilizer causes the roots to grow. Plotted on the x-axis. Your dependent variable, the effect. The response to one change is made. For example, the number of flowers affects the effect of the fertilizer is to produce flowers. That's plotted on the y. So we would have the amount of fertilizer on the x and the growth of the plants on the y. This is what I initially test. These are the results. So your results are going to be on the y-axis. For example, grasshoppers are exposed to different amounts of sunlight to compare how high. You have the y-axis and you have the x-axis. Let me go ahead and label those. X, Y. So which one's going to be on the X? What's going to be on the Y? You have the amount of light is going to be here, and how high the effect of the light is going to be on the Y. Next example. Students listen to different types of genres of music while taking a test to see if it improves the score. You would have the different types of music here, and then you would plot going on the y-axis their different test scores. This is what is occurring based on the experiment, what's on the X. And then your last one, plants are observed at increasing height after they are watered. So what's going to go down here on the X is the water. And what comes over here on the Y is how high they grow. Get it? Hopefully I didn't confuse you too much. If you're still confused, come see me. Okay. During a lab, you have these things that are called constants. And they're variables that remain the same in each of the experiment. A constant and something that is constant. It doesn't change. It doesn't move. Here's some, let's see if we can come up with some constants. Eels are placed in a dark cage and light cage. They are found to have slower swim speeds in the dark. Constants would be, would the eels be in the same cage? Yeah. Would they have the same type of water? Yes. Would they receive the same amount of food? Yes. Those are things that are constant in the experiment. That's not too bad, is it? Control. Part of the experiment that does not receive the experimental treatment. Not all experiments have a control setup. And here's an example. Create one group of rose plants that does not get a fertilizer at all. So you're controlling 1 spoon of fertilizer, 2 spoons of fertilizer, 3 spoons of fertilizer, and you're measuring the plant. Well, you can obviously say, well, are the plants going to grow anyway? How do I know if they're going to grow anyway? So on one plant, you put no fertilizer. Now, all of them are going to have the same pot, the same amount of water, the same type of soil. So those are all the constants. And then the control is the one thing that changes. Well, you're changing that variable, right? So that first plant that has nothing in it, that's going to be your control. In math, you have, I believe, Dr. ID to help you remember independent and dependent. In science, we use dry mix. They're the same thing. If you prefer one or the other, I'm indifferent. I taught math for a couple of years, so I think the Dr. ID is easier. And then last but not least, number the following 1 to 5 in their correct sequence. So what are we going to start with first? Right? We're going to state the problem. Maybe if I'm not writing in white, state the problem. Then what am I going to come up with? My hypothesis. Then I'm going to do my procedure. Right? I'm going to collect my data in the procedure, and then in the end, I will have a conclusion. That wasn't too bad. I hope I didn't put you to sleep. If you have any questions, please let me know. Have a good evening, and I will see y'all in a few days, hopefully tomorrow. Take care. Bye.